This short video is about the research pipeline and the broader idea of reproducibility and replicability, which is something that's going to be very important to us in data science. Okay, so from a research point of view, we can start by looking at this little chart. This is a chart by Peng and Eckel, and I'll link to this article from the webpage if you want to read it yourself. But the idea is, if we think about how data analysis works, how the research process works more broadly, even how the scientific method works, if you want to think in the broadest possible terms. The idea is that we go out and data is collected, and that's the blue box here. So data comes to us from some source. And unlike most science courses, which spend a lot of time thinking about where the data came from, how we collect it, how do we plan collecting that data, this course just assumes data has magically appeared before us. It could have been generated automatically by a machine. You could imagine all the sales data from various stores is maybe even calculated automatically from the cash registers. So it's not as though a human had to specifically take the data and give it to us. Or you could imagine it was collected by field researchers. So maybe there's survey research. So someone went out and he gave a survey to a whole bunch of people. Or a biologist was doing ecology out in the field. So she was out in the forest measuring insects or birds or plants or whatever it is she's measuring. And she has collected a data source. And what we know will happen is that raw data, the blue box, will be turned into analytic data, which is the first green box. And the idea of the analytic data is that it's been somehow processed or cleaned and made ready to be analyzed. And of course, that's a big chunk of what we'll be doing in the first next few weeks as we think about how we move forward with data. From that, we then do some analysis. We generate models, we generate statistics, and we get computational results. Often that's not in a form that's very pretty. Sometimes we go and we do some rough and dirty things to understand what's going on, and then we go back and make pretty ones later. The work you've been doing with ggplots would be a sense of that. But at the end, we would have some outputs. So some figures, some tables, some numerical results, some ideas. Then some text is written, often not by the data itself, but by a person. And the person has then generated some text, which they then include with the figures and tables and statistics or whatever to get an article or a report. So you could imagine from a scientist perspective, we start here and go here. But from a reader's perspective, we start with the orange boxes and we just try to imagine how we go backwards. So this idea that data exists, and at the end, a report or meaning or recommendations or an article is the output. As data scientists, as statisticians, sometimes all we're doing is the green boxes here in the middle. So we're given raw data, we spit out some things, and somebody else does it. You could even imagine a case, maybe we're working with that biologist, and when I do consulting, this is the sort of thing I do all the time. She gives us data, we do stuff to the data. When we spit it back out, we give figures, tables, numerical results, some raw text, some raw ideas, and she, the researcher, is going to turn that into the article. So what we're focusing on for most of this class is just the idea of how we get from measured data to analytic data, how we get results from that, and how we generate output that can be useful to the researcher, and might be us and might not be. So one idea that I want to talk about very particularly is the idea of reproducibility and replicability. Replicability, we're not going to talk about too much in this course, is the idea that if the experiment were repeated start to finish, we would get the same answers that we got at the beginning. If you imagine how research often works, often somebody doing a PhD level research, they're going out and finding some novel new thing to examine, to collect data on, to do this whole process start to finish. And that's great. For a master's thesis, often what you're doing is you're replicating another result. So maybe someone did find a really cool scientific conclusion. This new medicine works in this particular subgroup of people, and you're going to try it in a different subgroup of people, or you're going to try it in a broader range, or we found that insects tended to follow these patterns in this kind of a forest, and you're going to look in a different forest. So maybe the original research was done in the Pacific Northwest in Oregon, and we're going to reproduce that in forests in Missouri and see if insects behave the same way. So replicability is very important. And like I said, it's not really the thing we in data science worry about. What we worry about is the idea of reproducibility, which is that if somebody else is given the same blue box of data, the same measured data collected from whatever source, will they be able to, first of all, follow what we did so that it's very clear whatever decisions we made, we decided that it was normal enough that we could run a t-test, or that we should throw out that outlier because it doesn't make sense to include that outlier in our data, or we had to process the data because some of the dates were written as 4/15, 2015, others were written as April 15, so you had to turn the April into the 4. And so can we figure out how the process works start to finish? You could even imagine if we didn't show you what you did, would a person make the same decisions and would they come to the same conclusions? But reproducibility in particular is, will they follow that? Wikipedia actually does have a nice article about reproducibility. And so you can read that to learn a little bit more about that. But this idea that the process we use is very clear and clean. One word we use at the end of it is commensurate. So commensurate would mean that somebody who did the same thing would get the same output and they can understand where it came from. One nice thing about R is that the work that we do in R is very easily documented. In particular, the tools that come with R Studio, our Markdown, and the Tidyverse packages, which we're going to be using, make it very clear and easy for others to see. So for instance, when we were doing the ggplot lab, you were able to see how each step that we added, each line of code that we added very clearly made a difference in how the plot came out. We can add to this idea of reproducible research the idea of literate programming. And literate programming is the idea that when somebody looks at your code, they're able to read it, they're able to understand it, and preferably, even someone who has less computing skill than you or less statistical knowledge than you can figure out what you did and why you did that. When people talk about R, and especially R Markdown and the Tidyverse packages, they sometimes say that it's self-documenting code, meaning that the code itself explains what it's doing and it's really easy, right? When you say something like plus color equals green, it's easy to see that that's what made the color of the graph change to green. When we do the dplyr, which we'll do here soon enough about data cleaning, you'll also be able to see that it's very easy to follow when we make decisions about how to process a variable, exclude outliers, and do those sorts of things. It's very easy to use that idea. So that's the idea of literate programming and the idea of reproducibility. I do want to mention just one more thing about replication. This idea that replication should be done, again, we don't focus on it much in data science, but in the broader scientific community, it's been a huge thing. You can imagine for some things, replication is impossible. If we're looking at the sales data, from all of our stores, the sales data from all of our stores, we can't replicate it because we don't have another set of stores, right? We're not looking at a sample, we're looking at the population. Or data collection is so expensive that you aren't able to collect more data. There was a big study of great white sharks done a few years ago, and it had an N equals 14, so 14 sharks were measured. And you could imagine we statisticians would say 14 is too small of a sample size, you need more. And the shark people would say, man, do you know how hard it is to get data from sharks? So 14 is all you're getting. This has actually been especially an issue in psychology, where as they do start to replicate studies, they're finding that many of the results aren't replicable. So the second study doesn't find the same conclusion that the first one does. So much so that it's actually been called the replication crisis, which is the idea that if you can't replicate your results, then you shouldn't do that. So are there ways to build in more replication on the front end before you publish results or before you make a big deal about that? Of course, one problem isn't the scientists themselves, who might very well say in a research paper, this is preliminary data, we're not sure about it. I hope somebody replicates it before you draw too broad of conclusions. But maybe a media source or a web page or something just runs with the output, and that's where you get things like, I don't know, chocolate prevents cancer or whatever kind of weird things that you've been seeing. Okay. So that's the idea of reproducibility and replication, and this idea of literate programming. And in the coming weeks, we're going to be focusing on that a lot. So thanks.